Okay, class. So now we are back with understanding this last biological molecule, and these are the nucleic acids. What nucleic acids are? They are the DNA that we have and the RNA we have. As for this DNA, word has to be elaborated, so this goes for the deoxyribonucleic acid. And as far as this RNA is concerned, so this goes for the ribonucleic acid. In context of the RNAs, we further have got the messenger RNA, we have the ribosomal RNA, and we have the transfer RNA. But for your level, you need to be very much clear about the deoxyribonucleic acid, which is DNA. And among the RNA, your concept of the messenger RNA would be clear. Now, earlier when we were looking into understanding the structure of an animal cell, so I tell you guys that here we have the cell membrane, okay? And then we have got this nucleus coming, and in the nucleus there was existence of this DNA molecule. So we would look clearly at how we are required to be making this DNA molecule. So first we must know is that if this nucleic acid that we have, if they are the polymers, they are the large molecule, then what would be the small repeating units or monomers of them? So they are entitled as the nucleotides. As for the protein, we say amino acid, amino acid, amino acid, they combine to make the polypeptide chains. So likewise, when I have the nucleotides and combine with the nucleotides, so we would make the polynucleotide, and by the end we would make the DNA out of it. So if I am regarding DNA as a polymer, so the monomer of this DNA is a nucleotide with us. And as is the case with all the biological molecule, we must know that what elements we have got in the DNA. So it would be carbon, hydrogen, and oxygen. And we will have the nitrogen and as well as the phosphorus element. And now we must know that how come we are required to be making the DNA molecule. So I need to take a nucleotide. This nucleotide would be linked with another nucleotide, and from that we would make one strand of the DNA, and that one strand of the DNA is polynucleotide. And if I would combine two polynucleotide together, so we would end up making the DNA molecule out of it. Now this means that if we are not clear about the structure of the nucleotide, we at the end cannot make the DNA out of it. So you must know is that the small repeating unit or the monomer of the DNA has got three of the components in it. Like we would have one nitrogenous base, then we would have one phosphate group, and then we would have one sugar. When I would combine these three things together, so we would end up making up the nucleotide out of it. And these nucleotides, when repeatedly they would be linked, so we would make the DNA out of it. Now let us try to look at what we have to know in context of the nitrogenous base. So wherever the word nitrogenous base is coming, so this is one of the component of the nucleotide, and they are so called nitrogenous because in them we will see that there lies this important element in them, and this important element is the nitrogen element. So that is why they are termed as the nitrogenous bases, and we have got total of four nitrogenous bases in the DNA. By name, it is A, T, G, and C. E goes for adenine, T goes for thymine, G goes for guanine, and C goes for cytosine. And in the exam, they would have a symbolic representation, like the examiner would make some kind of a block like this, and in these blocks they would write these words as E, T, G, or C. So this is like for a nucleotide, I need to take one of the nitrogenous base. The other thing that we will have in the monomer of the DNA would be a sugar, and this is a five carbon containing sugar, pentose sugar, five carbon containing. And lastly, the symbolic representation of the sugar, it looks like this. Then we have this phosphate group, and the phosphate representation would go like this. So if someone was to say that you make for me the adenine nucleotide, so I would say that okay, fine. Adenine nucleotide mean that I have to be combining the adenine nitrogenous base. So I have got this adenine nitrogenous base. Then I need to combine sugar, and then I need to combine this phosphate group, and here I have ended up making one adenine nucleotide out of it. If someone were to say that make thymine nucleotide, so here I have made this thymine nitrogenous base and I have to make the sugar, then I have to make the phosphate. So when I have made these three components, we have ended up making thymine nucleotide. So these nucleotides, they combine with the nucleotide, and ultimately we would make one polynucleotide or one strand of the DNA. And the important thing is that in between the nucleotide, there exists a bond, and that bond is entitled as the phosphodiester bond. Now, usually for the DNA, we have got the two strands. So here we have the one DNA strand, and here we have the second DNA strand. And if you were to say that if I have this one strand, this is the second strand. So in between the nitrogenous bases, there would be existence of some attractive forces, and these are the hydrogen bonds. And these nitrogenous bases within the one strand, they combine in a particular or in a specific manner to the nitrogenous bases that we have in the next strand. If I were to have adenine here, then in the second strand it must always be thymine. If I were to have a thymine here, so in the second strand there has to be adenine always. If I would have G here, so in the second strand the nitrogenous base should be C. So you must remember from a word that I will meet you at GC. So at mean adenine and thymine, GC mean guanine and cytosine. And this kind of the pairing that exists between AT and GC, so we call this as the complementary base pairing. So you see for the DNA, this is one polynucleotide or one strand. This is the second polynucleotide or the second strand, and collectively, when in these two strands, if these hydrogen bases, when they link up with one another or they combine or they kind of attract one another with the help of hydrogen bond, so we will have two strands, and these two strands they would make one DNA molecule. So now we must know that how come the DNA structure looks like. So you see that the DNA overall is in the form of a double helix. Here is the one strand, here is the second strand, and then we have the small repeating units in them. They are the nucleotides. The nitrogenous bases that we have in the DNA, so they would make the complementary base pairing. Adenine would be lining with thymine, the guanine would align with the cytosine, and in between them, there would be existence of these hydrogen bonds. Okay, so this is all you must know in context of the structure of the DNA. Now, what is the role of the DNA? Actually, is I have cleared it in the cell structures and the function video lecture that you can find it at the top. And over there, you will see that this DNA that we have in our body, this is running one important role that it would be making the proteins in the body. So this is here we are done with all the biological molecules.